All right. It is 7 a.m. in San Diego, California, and the sun is just about to rise out here outside of my window. It might look quite different for you, depending on where you are. We've got a really solid number of people signing up here for the webinar, really across the globe. So thank you for joining. Thank you for the interest in that topic. My name is Daniel Sauter. I'm an application science manager here at Sophion Bioscience. And I have the pleasure of hosting this event today on advancing organelle electrophysiology on lysosomal and mitochondrial ion channels. So I still want to give some time for people to come in. It's 1 minute over. And as we then go along, we can then start out with the topic of today. So as I mentioned, we have two exciting presentations lined up. First up, we will have our long-term partner at Axxam in Italy, Dr. Manuel Paina, who will focus in on mitochondrial ion channel. He's then followed by my colleague, Dr. Eliška Waloschková, who will give a short introduction to a collaboration that we had with a French company specializing on isolating lysosomes. And she will also introduce a new product line that we've developed, a new consumable, really that caters specifically to the needs of studying organelle ion channels and also small cells and other small vesicles. So before we start, a few housekeeping items. We use that Demio platform that we're quite happy about, and you'll see here on your right-hand side a chat functionality that I will invite you to use to post your questions throughout the presentations. We plan then to have a combined Q&A session at the end of the webinar when we go over them, but really that should not hold you back from posting your questions throughout the presentations. We are also interested in learning on where you see the field moving and really where you stand with organelle ion channels. What's your interest? For that reason, we're going to run several polls that you see just right next to the chat functionality. And I would appreciate if you participate in those polls. It is really valuable information that we can gain out of that. So lastly, we also have a handout section on the right side where you see some information about our products, the new developments that we're introducing here, but also some relevant lysosomal recordings posted that you can download from there. So this is really an exciting time for us to participate in that field of organelle ion channels. It's great to see for me. I'm with the company for now close to 10 years. And really during that time, I've seen how ion channel research has expanded beyond the plasma membrane and really embraced these ion conductances in the intracellular organs. And really, it's not limited to lysosomes that we hear a lot about. Ion channels play a critical role also in different other organelles, endoplasmatic reticulum, the mitochondria, as we hear later, the Golgi apparatus, endosomes, the nucleus, to name a few. These ion channels are really emerging as critical regulators of cellular homeostasis, regulating ion gradients, pH, obviously the membrane potential, trafficking, autophagy, nutrient sensing, and signaling. So importantly, dysfunction in these channels is increasingly linked to disease states, and prominently here is the involvement in various neurodegenerative disorders, but also metabolic disorders, lysosomal storage diseases, cancer, and even infectious diseases. These organellar ion channels play a role in here. So you could ask really, why do these organelle ion channels more come on stage or move into the center a little bit delayed compared with plasma membrane ion channels? And really, there's several reasons, and a big one certainly is the challenge that it poses studying these ion channels due to the small size of these organelles and the difficulty to isolate them. So what we will see here today is the introduction of different tools that really have allowed the field to really consider these ion channels more as druggable targets. And there are great examples out there already where, for example, the lysosomal proton and potassium leak channel, the TMM175, that is involved in Parkinson's disease, as well as the lysosomal calcium channel, the TRPM-L1, involved in lysosomal trafficking and autophagy, they are being actively pursued by the pharmaceutical industry already, and we see great progress being made there. So without further ado, I think there's a lot to learn in that field, and we are quite excited to host that webinar here today to introduce some of the advances happening. And for that, I mentioned we have two presenters available. The first will be Dr. Manuel Paina, who is joining us from Axxam in Italy. Briefly about Manuel, it's actually a continuation. We had Manuel here on the stage in 2021, where he showed a webinar that I really invite you also to check out in our YouTube channel, where he spoke about the early advances that Axxam did with recording or developing assays for different organelle ion channels. So Manuel briefly received his PhD as well as his postdoc from the University of Milan, where he first studied HCN channels and then moved on to focus on NAD 1.5 in his postdoc. He joined Axxam in 2018 as a scientist, became then a principal scientist, and was then in 21 promoted to the role of the supervisor in electrophysiology, where he specifically manages the assets around organelle ion channels. So in this role, Manuel has really been at the forefront of organelle ion channel assay development, and I'm really excited to have him here on the webinar today to learn more about his recent progress. So without further ado, Manuel, I want to give it over to you. I'm going to switch off the camera.
Thank you, Daniel. And so before diving into the topic of organellar electrophysiology, let me give you a brief overview about our company. So Axxam is a science-driven drug discovery company, and our mission is to help clients turn innovative target biology into high-quality chemical solutions. As you can see in the diagram, our expertise are related to the drug discovery processes. In particular, the Discovery Max package is a program starting from target ID and validation through hit identification, hit to lead, and lead optimization at the end. Axxam was founded in 2001. Our headquarters and biology labs are based in Bresso near Milan, while our chemistry labs are located in Torre Annunziata near Naples. We have also an international presence with offices in Cambridge, US, Copenhagen, and Basel. Our business is related to several sectors, from pharma and biotech to agrochemicals, flavors and fragrances, and even food and beverages, making us a truly cross-disciplinary partner in innovation. So when I introduce the Organellar Electrophysiology, I like to start with a simple question. Do you know how many transport processes occur within intracellular organelles? And the answer is surprisingly, because it's around 80%. 80% of all transport processes take place on ion channels and transporters located on the membranes of intracellular organelles. And while the physiological role of plasma membrane ion channels and transporters are well studied and characterized, the intracellular counterparts remains far less understood. In addition, organelles ion channels represents attractive drug targets due to their unique sequences and three-dimensional structures, which would theoretically allow the development of selective and specific drugs with fewer off-target effects. At Axxam, we have developed a series of dedicated assays designed to investigate ion channels specifically expressed on organelle membranes. These include the NucleoPatch for nuclei, Mitopatch for both mitochondria and mytoplast, and Lysopatch for lysosomes. These innovative approaches allow us to explore organelle-specific ion channel function in a native-like environment, opening up new opportunities in both basic research and drug discovery. Our results have already attracted attention and have been presented at major scientific conferences, such as the Biophysical Society meeting in 2020 and 2024. At Axxam, our journey into organellar electrophysiology started with a white paper and a clear goal: to develop a series of assays that could meet both scientific demands and customer needs in this emerging field. The first step in this journey involved the use of manual patch clamp techniques, where we recorded single-channel activity from ion channels expressed on the nuclear membrane and studied how their activity could be modulated, as you can see in the panel 1. The next step was to develop the Mitopatch, a dedicated assay for the characterization of mitochondrial and mitoplast ion channels. Here, for example, in the panel 2, you can see a single-channel recording from a mitochondrial permeability transition pore, a key player in cell death and mitochondrial function. A significant portion of our work has also focused on lysosomal electrophysiology. We have established multiple experimental assays for studying ion channels in lysosomes, using both manual and automated patch clamp platforms. This versatility allows us to adapt to a wide range of research needs and compound screening workflow. Today, we are also pushing the boundaries of the field by recording ion currents from lysosomes isolated from iPS-derived neurons, such as motor neurons and dopaminergic neurons. This work is part of an ongoing grant project called ICOS, which focuses on the development of in vitro assay platforms to identify new pharmacological compounds for the treatment of neurodegenerative diseases, in particular ALS. And last but not least, we are now applying automated patch clamp technology, specifically the QPatch platform, to isolated mitoplasts. This approach allows us to record the activity of specific mitochondrial ion channels and develop robust assays for their detailed functional characterization. This is the main focus of my presentation today, and I'm excited to share with you how this platform is helping us to explore new therapeutic targets within organelles. First of all, I'd like to begin with a brief introduction to ion channels, and more specifically their role at the mitochondrial level. Within mitochondria, ion channels are located on both the outer and inner membranes, and they play a central role in maintaining cellular and organellar homeostasis, as well as regulating several key cellular processes. Among their most important functions, mitochondrial ion channels are involved in the regulation of membrane potential, control of reactive oxygen species release, volume regulation of the organelle, supporting the function of the respiratory chain complexes, and mediating intracellular signaling pathways. In the last years, a number of new ion channel proteins have been identified on mitochondrial membranes, revealing even more complexity and functional relevance. These discoveries have advanced our understanding of mitochondrial physiology and its integration with overall cellular functions. As shown in the cartoon of the slide, some of the most studied mitochondrial ion channels include the mitochondrial calcium uniporter MCU, the mitochondrial permeability transition pore MPTP, BK channels, and TASC3, among others. These channels are not only fundamental to mitochondrial activity, but also represent promising therapeutic targets in the context of several diseases. When we talk about therapeutic targets, mitochondrial ion channels are gaining increasing attention, and for good reason. Malfunction in these channels are often associated with a wide range of cell-originated diseases, as illustrated in the image, which highlights their impact across multiple body systems. This dysfunction can disrupt intracellular communication, leading to impaired cellular homeostasis and, in many cases, contributing to the development of neurodegenerative, metabolic, and cardiovascular diseases. Once these channels are properly identified and characterized, they become attractive pharmacological targets, and we can now introduce the mitochondrial medicine, an emerging and highly promising field. By designing selective modulators, we can intervene at a very early stage of the disease process, potentially restoring normal function of the mitochondrial level. Restoring mitochondrial function is not just about fixing a single organelle. It could represent a transformative approach for treating a broad spectrum of diseases where mitochondrial dysfunction is a key driver. Organellar electrophysiology is a powerful technique, but traditionally it involves highly focused and low-throughput experiments due to the technical complexity of working at the organelle level. The introduction of automated patch-cam platforms such as the QPatch is a real game changer. It allows us to significantly increase the throughput while maintaining the high-quality data, and this is essential in this field. If we look at the funnel of experimental approaches, the QPatch represents a bridge between low-throughput manual patch clamp and higher throughput screening technologies. And this is exactly the gap that the collaboration between Axxam and Sophion aims to fill. However, the development of automated organellar patch clamp has several challenges. First of all, obtaining highly purified mitochondria is critical. And without clean preparations, automated experiments become unreliable because you cannot visualize the proper organelle during the experiments. Second, we needed to work with customized QPlates, specifically optimized to handle small organelles like mitochondria and lysosomes, allowing efficient capture, sealing, and stable whole organelle recordings. Finally, one of the key steps was to improve the experimental stability, in particular maintaining the sear resistance throughout the duration of the assay. This required extensive protocol optimization and condition screening to ensure consistent and reproducible results. This advancement opened the door to applying automated electrophysiology to previously inaccessible targets, and this is a major leap forward for both basic research and drug discovery. At Axxam, we have developed a customized protocol for mitochondrial isolation, starting from a commercial kit, but significantly improving its performance to meet the specific requirements of organellar electrophysiology. To validate this protocol, we carried out a series of imaging experiments using the Operetta high-content screening system. As a first step, cells were incubated with MitoTracker, a fluorescent probe that is commonly used in cell biology to visualize mitochondria. This probe is made up of a lipophilic cationic dye, which can pass the plasma membrane of the mitochondria and live cells, with accumulation due to the high membrane potential across the mitochondrial inner membrane. We then proceeded with the isolation protocol, and at the end we obtained what we call a mitoprep, a preparation highly enriched in intact mitochondria with minimal cellular debris. An additional step can be introduced according to the experimental needs, a thermal shock applied after the isolation. This treatment disrupts the outer mitochondrial membrane, allowing us to obtain mitoplasts, which are mitochondria without outer membrane, ideal for electrophysiological recordings focused on the inner membrane. Images of the resulting mitoprep were acquired using the Operetta system, as you can see on the bottom part of the slide. Here you can see two representative fields and a magnified view of one of them. The colored dots indicate individual mitoplasts, and the table on the right summarizes the number of objects detected, confirming the efficiency and consistency of isolation protocol. For the QPatch assay development, we used our experience with the manual patch clamp. One of the most important things is the shape and the quality of the pipette tip, which plays a central role in achieving high-quality recordings. In manual patch clamp, we use a microforger to polish pipettes to smooth and refine the tip. This process reduces background noise, improves seal formation with the membrane, and ensures greater recording stability. A well-polished pipette is essential to obtain gigaohm seal resistance, which is critical for accurate and reliable measurements. A similar approach has been adopted by Sophion in the development and customization of the Q plates, the key consumables used in the QPatch system. These plates are engineered and include a silicium chip as substrate to enable gigaohm seal without the use of any seal enhancer, microfluidic flow channels that allow rapid and complete solution exchange, and customizable hole size, which are critical when working with non-standard samples. For our organelle-specific assays, special organelle-dedicated couplets were produced featuring reduced hole sizes. This adaptation ensures efficient capture and sealing of small organelles such as mitochondria and lysosomes, enabling high-quality whole organelle recordings in an automated format. More in detail, the engineered single-hole couplets were specifically designed to enable reliable capture and gigaseal formation on isolated organelles. In the middle panel, you can see a screenshot from a representative experimental run. The color coding indicates the quality of the seal. Yellow marks successful gigaseal, while blue indicates position where sealing was not achieved. In this specific experiment, we obtained 3 gigaseal out of 16 positions, all without the use of any seal enhancer. This means the seal were formed entirely under physiological conditions, demonstrating the robustness of the system. It's also important to note that the blue dots do not represent failed attempts on viable mitoplast, but rather position affected by debris or poor capture. This distinction highlights both the precision of the QPatch design and the importance of sample quality in this type of measurement. Additional point, as you can see in the table on the right, the capacitance values measured by QPatch are consistent with the small size of mitoplast, and this is very important because it allows us to properly calculate the current density. So the last part of this presentation focuses on a case study centered on the mitochondrial calcium regulation. Calcium ions play a crucial role in numerous cellular processes. Intracellular calcium is stored and regulated across various organelles, including the ER, mitochondria, Golgi apparatus, and lysosomes. This signaling relies on a complex network of channels and exchanges embedded on organelle membranes. In our study, we investigated the mitochondrial calcium uniporter, or MCU, using isolated mitoplasts and the QPatch. The MCU is a highly selective calcium channel located in the inner mitochondrial membrane. It's composed of a pore-forming core subunit and several regulatory subunits. Physiologically, the MCU plays a pivotal role in shaping intracellular calcium signaling, regulating aerobic metabolism, and triggering apoptosis. It allows calcium to enter the mitochondrial matrix by following the electrochemical gradient. The activity of the mitochondrial calcium uniporter is tightly controlled by regulatory proteins, which act as gatekeepers based on cytosolic calcium levels. From a pathophysiological perspective, disruption in mitochondrial calcium homeostasis have been linked to a variety of diseases. These include neurological disorder, cardiovascular conditions, and several forms of cancer. Notably, altered expression of function of MCU components have been associated with tumor progression and cancer-related phenotypes. For this case study, the material and methods are reported in the top panel. We used the whole mitoplast patch clamp configuration to investigate the activity of the mitochondrial calcium uniporter. As shown in the cartoon, the experimental setup includes a Q plate with an isolated mitoplast captured on the top of the recording chamber. The voltage protocol applied was a ramp from -80 to +80 millivolts with a duration of 400 milliseconds, starting from holding potential of 0 millivolts. We used a single-hole, high-resistance Q plates. Their high resistance minimizes leak and noise, improving the quality of the recordings. Regarding the solutions, the intramitoplast solution is similar to the intramitoplast environment, and it was potassium-based, while the external solution, which mimics the mitochondrial matrix, was Hippes buffered. Both solutions were adjusted to a neutral pH to maintain stable and physiological recording conditions. The application protocol was carried out in several steps. Initially, mitoplasts were maintained in a calcium and magnesium-free solution to establish a baseline with no MCU activity. This was followed by multiple application of the external solution to fully remove the previous buffer. Next, we applied a compound at single concentration to observe its potential effect on the mitochondrial calcium uniporter current. Finally, we use a full block, in this case containing 20 micromolar of ruthenium red, a known and potent inhibitor of the mitochondrial calcium uniporter. At the bottom of the slide, you can observe two representative traces. The gray trace corresponds to the basal current in the control condition before calcium application. The blue trace shows the response to 30 micromolar calcium, which activates the MCU current, and the red trace represents the complete inhibition of the current after the application of ruthenium red. This stepwise approach allowed us to confirm the specificity of our current and the effectiveness of both the activator and the blocker within our experimental system. After this experimental validation, we started to develop different assays to address specific experimental goals. The first assay was designed for single-point screening, where each compound is tested at a single concentration. This approach is useful for a quick comparison of multiple compounds' effect under standardized conditions. In each experiment, after the compound application, we applied a specific full block to validate the observed effect. In the experiment presented here, we tested 4 different compounds. We measured the current amplitude at -80 millivolts as a readout of MCU. These data were then analyzed and compared across the compounds. The results are summarized in the bar graph, where we calculated the compound-induced current considering the basal channel activity recorded during the vehicle liquid period. We can clearly observe which compound have stronger activating effects, providing a basis for the further functional characterization. A different assay was then developed to perform dose-response experiments, aiming to determine the compound's potency and generate a complete dose-response curve. After preliminary tests, we selected 4 increasing concentration of the compound under investigation to cover pharmacological range. In these specific experiments, we evaluated camphorol, a compound reported in the literature as a selective activator of mitochondrial calcium uniporter. The concentration tested were 3, 10, 30, and 100 micromolar, chosen based on previous studies to ensure compatibility with known data. On the right side of the slide, you can see representative traces of mitochondrial calcium uniporter current recorded in control conditions and after the application of increasing concentration of camphorol. These traces show a clear trend of current enhancement as the concentration increases. The collected data points were then used to create the dose-response curve from which we calculated an EC50 value of 9.3 micromolar, indicating the concentration at which this compound elicits half of its maximal effect on MCU activation. To conclude, The assay development has led to the successful optimization of dedicated QPlates specifically designed for use with isolated organelles such as mitochondria and lysosomes. These plates now enable efficient capture and seeding, making it possible to perform recordings in a whole organelle configuration, which is critical for studying the native activity of ion channels in their physiological context. The QPatch platforms has proven to be a highly effective system for this type of application. It supports high-throughput recording with up to 48 parallel experiments and consistently delivers high-quality electrophysiological data characterized by gigaohms resistance and reliable cell capacitance measurements. Overall, QPatch represents a valuable tool for compound screening, also for ion channels localized on organelle membranes, a field traditionally characterized by significant technical challenges. In addition, the platform supports the generation of dose-response curves, allowing researchers to determine key pharmacological parameters such as EC50 directly within the native environment of the ion channels, thereby increasing the physiological relevance and predictive power of the data. And then I leave the floor to Eliška for her part. Thank you.
Well, thank you very much, Manuel, for this really great presentation. I mean, the 4-point cumulative concentration response curve on camphorol is definitely a highlight for me there. All right, so with that, I want to remind everyone quickly to post the questions here in the chat. We'll take all of the questions then at the end of the session when we invite both of the speakers on stage again. But now we have our second speaker, and it's my pleasure to introduce Dr. Eliška Waloschková. Briefly about Eliška, she received her PhD from Lund University in Sweden, where her PhD project that was funded by a Marie Curie scholarship was on developing new gene and cell-based therapy approaches for treatment of drug-resistant epilepsy using different stem cell models. She had a short stint at AstraZeneca then before she relocated to Copenhagen to complete 2 postdoctoral trainings at the University of Copenhagen, first focusing on Alzheimer's disease and then later on developing innovative bioluminescent mouse models. So she joined Sophion as an application scientist in 2024 and has since done great work also focusing on lysosomal electrophysiology. So without further ado, Eliška, the stage is all yours.
Hello, and thank you for the nice introduction, Daniel, and thank you, Manuel, for a great presentation on MitoPatch using the QPatch. So I will tell you a little bit about the lysosomal patch clamp using the Sophion systems. And as Daniel already mentioned in the beginning, this is a collaboration with a French biotech company called Organellar Bioscience. So what are lysosomes? So just a quick introduction. Lysosomes are one of the many organelles inside the cell, same as mitochondria, nucleus or the endoplasmic reticulum. They are much smaller than a cell itself. They contain acids and digestive enzymes to break down macromolecules, such as proteins and carbohydrates. And they are also able to destroy foreign particles like bacteria and viruses and protect us from that. And lysosomal ion channels are, of course, involved in all these lysosomal functions. They regulate the internal pH to be able to digest macromolecules. They manage the storage and release of, for example, calcium. They control membrane fusion. And the dysregulation of these ion channels can lead to cell toxicity and diseases. It's involved in neurodegeneration, some rare diseases, and cancer. So of course, lysosomal ion channels are then a great target for drug discovery. But organelles are small in size. So how do we measure these organellar ion channels? And for this, as Manuel already mentioned, we have customized our consumables. And that is the small hole Q-chip. That is for our Cube platform. That is our high throughput 384 channel platform. And the Q-plate, which we call the high resistance Q-plate with a small hole, which is then used on the QPatch. So a 48 channel platform, but we also have a smaller Q-plate available for the QPC, which is the QPatch Compact. That is an 8 channel platform. And as I mentioned, this is a collaboration with Organellar Bioscience. So Organellar provides us with ready-to-use lysopreps that we can use with these small hole consumables to record lysosomal ion channels on our systems. So we started the assay development on our high throughput platform, the Cube. And our first aim was to validate that we are able to capture lysosomes onto these smaller Q-chip holes. And you can see this on the slide where on the panel on the left, in the first 4 rows of the Q-chip, we did not have any lysosomes in our solution. So this was a negative control for us. And then the rest of the Q-chip we filled up with lysosomes, and we can see if a lysosome gets positioned onto the Q-chip hole with looking at the resistance plot, which you can see on the right side. And if our resistance rises suddenly, means that there's a lysosome being captured on the Q-chip hole. So this was the start of our lysosomal assay development. And we also confirmed that the small hole consumables reduce lysosomal slipping. You can see this looking at these 2 resistance plots. On the left side, you can see the resistance going up and down, meaning there's lysosomes flowing through the patch hole and not getting captured. While on the right side plot, you can see that the lysosome gets captured on the Q-chip hole. So regarding the consumables on the Cube, we use the small hole Q-chips. The hole there is around 1.5 micrometers small. And we started this assay development with standard Sophion solutions, which we know that work really well on our systems. And this was a really good starting point for us. We managed to achieve good lysosome capture rates. Here you can see on the left side whole Q-chip screenshot where the green sites represent lysosomes that passed our QC criteria. And we also managed to record nice currents from these lysosomes, which you can see on the right side. And now I will delve into a specific ion channel that is specific for lysosomes, and that is the trypamol-1. And just to quickly introduce you to trypamol-1. So trypamol-1 is a non-selective cation channel, so it's permeable for calcium, potassium, sodium, and so on. It's involved in a lot of lysosomal functions, including exocytosis, autophagy, homeostasis of iron, but also degradation of macromolecules. And why is trypamol-1 important? Is that mutations in this channel cause mucolipidosis type 4, which is a severe neurodevelopmental disorder, but also its dysregulation is linked to neurodegeneration, metabolic disorders, and cancer. So for this, we switched to the QPatch, the QPatch 48, and we had to really study this ion channel to then come up with a solution recipe for TRPM01, where the important was to change the ion composition, but also keep the luminol solution at the acidic pH. So just a tip for your assay development, it's really important to know your ion channel. And for the QPatch, we managed to record around 60% of successful experiments. And we also confirmed that we are actually recording a TRPM01 current by using agonist called MLSA5, which you can see down in the bottom right corner with the blue trace. We also managed to record cumulative concentration response experiments, as you can see here, and we measured the EC50 of MLSA5 to be around 0.25 micromolar. And just a little teaser, we managed to now transfer this assay outside of Sophion, and our other collaborator, BSIS, managed to record these lysosomes on their QPatch system as well, and also do cumulative concentration response experiments, and measure the EC50 for MLSA5 at 0.50 micromolar. We then tried to transfer this assay to the Cube, so to our high throughput platform with 384 channels. There we have around 40% successful experiments, and we also confirm the TRPM01 current by using the same compound, MLSA5, and again you can see this activated trace on the right side here. So to summarize, we are able to capture lysosomes on our small hole consumables. On the QPatch, we have around average of 70% lysosomal capture. On the Cube, it's around 65% of lysosomal capture. And again, I have to remind you that this then depends on what solutions you are using on the systems. And we are able to target high-profile ion channels, such as TRPM01, which we confirm with pharmacological activation. And with this, I thank you for your attention and go back to Daniel.
Thank you very much, Eliška. Very nice presentation, great overview, and great data. We do have a number of questions, so I do invite Manuel also on stage again, then we can have a panel discussion going over the various questions that we have here in the chat. So starting from the top, Heike, hey Heike, has a question asking, have any of Sophion technologies been used for isolated channel protein reconstituted in bilayers? I guess, Eliška, maybe you want to take this question?
Sure. We do have a collaboration to work on this. It's a work in progress. So I don't have any data to show yet, but I can say that it is something that we are working on, specifically on the QPatch at the moment.
Yeah, that is exciting. So we have the next question comes in from Matthew. Have you studied trip channels on mitochondria? If yes, like to hear the technical difficulty and reliability of the results. Manuel, I think that's a question for you.
Yes, so at the moment we have experience with different mitochondrial targets, and in particular at the manual patch clamp platforms, we work with the mitochondrial permeability transition pore, and I show during the presentation single channel recordings and also mitochondrial calcium uniporter. While at the QPatch, we are at the beginning of the assay development phase, and we decided to work with mitochondrial calcium uniporter because there is a really good activator and blocker. But we have in mind additional targets for the assay development, and trip channels are for sure one of them. So in the future, we have the intention to work with this target.
Fantastic. We have a question coming in from Christina. What is the % positive cells using the QPatch technology? I guess that's a question really for both of you. So Manuel, why don't you touch on that first?
Yes, so the percentage of successful experiments using isolated mitochondria at the moment is about 20–30%. So I know this is not a large percentage, but we have to keep in mind the technical difficulties of this technique. And also we have to compare this percentage with the low throughput of the manual patch clamp. So at the moment, I consider the QPatch 48 a real game changer in this field.
Yeah, that is such a good point. If I just also can sort of touch on that, because I think that's so often been overseen. Like when you represent those 20–30% for people in screening, they often look at it and say like, hey, you know, what about the other 70%? But I think you make a very good point there that you really have to compare it to the manual patch clamp technique. And you come in with a fast acceleration of your throughput using such technology, even with the comparably low throughput compared with a recombinant cell line. But yes, I think that's a great point. And I think also for you, especially showing that you use physiological solutions there, which of course like makes it even more challenging to get good recordings out. And I think that's why the 20–30% is quite impressive actually. Eliška, do you also want to touch on that question?
Yeah, sure. So for the QPatch specifically, we managed to get around 60% success rates. And this is with specifically the Oraya lysopreps. So it's a standardized product that we are using on the QPatch.
Nice. Yeah, all right. So let's move on to the next question that comes in from Brian. So does the QPatch platform require any upgrade to run these new Q plates? Eliška, do you mind taking this one?
So I have to say yes and no. So you can run these high resistance Q plates without having any upgrade. There will be an upgrade regarding measuring capacitance values, where, for example, for a specific type of protocol, our capacitance values for the really small organelles falls below a threshold that the system cannot measure. But as it is now, you can use the high resistant Q plates without any upgrade. It just depends on then how big are your organelles and if you are using a timed or non-timed protocol. So it really depends on your application. But you can use them, and if you want to, then just come in contact with us, and we can help you with setting up your assays.
Exactly, certainly. We are here to help if you want to try out lysosomes or any other organelles on your system. Moving on to the next question that comes in from Peter. Very impressive presentations. Any progress made so far using the high resistance consumable plate for ER or ER nuclear isolation? Manuel, why don't you take this first?
Yeah, at the moment we are focused on lysosomes and mitochondria. So I well know the importance of ER, and it could be the next step. And just to be clear, in Axxam, when we start to develop a new assay, we started with manual patch clamp platforms to validate the target and the assay, and then we move on automated patch clamp platforms. So at the moment, we have an idea to move on ER, but there are no results for now.
All right. Eliška, do you also have a comment to that question?
I just have to say we haven't tried yet, but we are interested in doing so.
Yeah, so I mean, there's a related question, really, the next one coming in from John here. What do you have for the ER and how does it compare with the optical patch readout? I guess we've kind of touched on this then. I don't know, any further remarks there, Manuel, from you, or? Yeah, I think we probably touched on it in the first round there with your answer. I'll move on to Maya with the next question. So, is it necessary to modify the suction pressure for stable gigacell formation on lysosomes? Eliška, why don't you take this one first?
Yes, very good question, very specific. And I have to say, yes, you need to modify your wholesale protocols to be specific for your lysosome solution. So then that has to be optimized as for any other, as for a normal cell line. If you are starting an assay with a new cell line that you haven't done before, probably you will need to adjust your strength of the pressure, both the suction or positioning pressure. So, of course, there are some adjustments to be made for the lysosomes.
Nice. All right, we have the next question coming in from Vadim. Thank you, very interesting presentations. Did you try more than 4 concentration points for the dose response curves? So Vadim's idea here is that there could be subpopulations of channels, etc. Manuel, I think that's for you.
Yeah, at the moment, so we decided to work with 4 concentration, just to have a protocol not so long in order to maintain a stable sealing during all the experiments. But for sure, when we are able to optimize more our assays, we can introduce an additional liquid period in order to have additional point to create a full dose response with a larger number of points. But at the moment, we decided to work with 4, that it's a good compromise to cover a full range of concentration.
All right. Yeah, very good. I mean, I think these cumulative those response curves, I think that really is that really did impress me, because I think it really made use of the microfluidic flow channel, where you can really sort of like bring in new concentrations and really sort of like utilize your recording to the best possible way to get most data out of it. I'm moving on to a question from Mark here, who says, very nice data, tantalizing process for sure. It's a question for Manuel at Axxam. It looks like you screened for MUC, MCU probably activators, in the absence of activating calcium. Is that correct? And if so, why?
Yeah, so I didn't report the full composition of the solutions, but we have a percentage of calcium in our solution, just to maintain the activity of mitochondrial calcium uniporter. So we have calcium in our solution, yes. And then we have also camphorol to evoke an higher activation. But yeah, the answer is yes, we have calcium in solution.
Okay. Very good. A last question we have here from Michaela. Have you ever tried lysosomes from other isolation techniques? Manuel, I think if you want to go first on this one.
Yes, we in Axxam we have our specific isolation protocol for lysosomes. So again, it's customized in our labs and it's created to obtain a lysoprep, in this case with high concentration of lysosomes. And so we have huge experience with this protocol because it allows to perform experiments using the automated patch clamp platforms.
Yeah. All right, fantastic. Eliška, any further comment from you?
I just have to say that at Sophion we have not tried any other lysosomes than the Ori lysosomes, which work really well for us. So.
Yeah. All right. We do have a follow-up question coming in from Mark that is for you, Eliška. So to follow up on the wholesale protocol question, are these new organelle protocols going to be available in the Hive database? Quickly, for those of you who don't know our Hive, we have just rolled out the Hive, which is an online platform that really makes use of our 25-year history developing assays for automated patch clamp technologies. We really combined all the in-house data here and used advanced algorithms to allow to quickly extract the best performing wholesale protocol for a given cell type, for a given consumable, for a given ion channel. So that's what is being referred to here as the Hive database. Please check this out. And now, Eliška, I want to give you the question here, whether the lysosomal protocols will be available in Hive.
Yes, that is a good question, and I believe they can be made available. However, the protocols that we use at the moment are timed. That means that the success rate that the machine tells you is always 100%. So I would say in Hive, it wouldn't be super clear which one of them is the best protocol, since they will all have a 100% success rate. So I think if you want to discuss the wholesale protocol, you can just reach out to us. But yes, they should be at some point available on the Hive database.
Okay, very good. Brian has another question. I may have missed this, but is the high resistance QPlate using single hole or multi-hole mode, Eliška?
It's a single hole. It's a single hole recording. Yeah.
Yeah. All right. I think with that, we went through. I have to go over here to see if nothing new came in. No, it seems like we have gone through the questions here. I want to also point out here really quickly that whilst you've seen really some of the current developments that we have going on at Sophion, stay tuned. We have more developments in the pipeline that we'll expect to come out with next year, specifically for the QPatch technology, where we optimize different elements to improve that technology, in particular the amplifier. I think for those of you who paid attention, Eliška really showed, and Manuel as well, showed some recordings that really had really small amplitudes, which sometimes these currents aren't really expressed at high levels. So having like a very sensitive amplifier available is a key advantage. And stay tuned for more on that topic. We will have some exciting news to share with you on that front as well to further improve these organelle recordings. Well, now my German heart is jumping in excitement because I see we are finishing just on time. So I want to use that last minute to thank the presenters again. Thank you, Manuel. Thank you, Eliška, for joining us here on that webinar. And then, of course, all of you for joining this webinar and asking good questions. Please feel free to reach out to us in case something is left unclear at this point. But with that, I'm closing the session. Thank you.